Hello friends, welcome again to our new video. In the previous video we have talked about the equipments used in our pathological or microbiological laboratories. So I have provided previous class link. Those who have missed can go to this link. And also I would like to mention that if you have any doubt, you can use every way for communication. Like you can use comment box, you can message, you can mail or WhatsApp, etc. So before moving to today's lecture, I would like to remember a few important points which are related to our previous class. Equipment hot air oven. It is developed by Louis Pasteur. Autoclave is invented by Charles Chamberlain in the year 1879. Abbreviation of HEPA, that is called as HEPA filter, is high efficiency particulate air. HEPA filters removes 99.9% of all air particles which have diameter more than 0.3 micrometer. The Petri plates are named after German bacteriologist Julius Richard Petri. So these were a few points I wanted to share with you. Moving to today's class. So first, I would like to share some terms which are going to be used in this lecture. First is pure culture. What do you mean by a culture? A culture, it is one which only a single known species of microorganism grown. You can see in this picture only a single microorganism is grown, so it is called as pure culture. It is also known as axenic culture. So a pure culture is a population of cells derived from a single cell. So next is mixed culture. These are the culture that holds two or more identified and easily differentiated species of microorganism. As this picture shows, growth of two different organisms, you can easily differentiate them. Another term we can use is contaminated culture. So the name itself suggests contamination, the growth of unwanted microbes in a culture are known as contamination, or the culture is known as contaminated culture. So here we also should know what culture media is. A culture media is the substance which provides nutrients for the growth of microorganisms. For example, potato dextrose agar, it is used for growth of fungal microorganism. The nutrient preparation on which culture is grown in the laboratory is called culture media. This is another definition. The growth of microorganisms on culture media is called culture. Like a culture, a colony of trichoderma can be called as a trichoderma culture. So moving ahead, I would like to discuss about types of media. So types of media, we can classify or differentiate different type of media on many aspects, such as based on chemical composition. A media can be classified three categories. First one is natural media. Next is synthetic media, otherwise synthetic media. So as the name itself suggests, natural media. In this media, the exact chemical composition is not known. Like we use carrot agar, oatmeal agar, rapeseed agar, etc. In the synthetic media, the exact chemical composition of media is known. So they are called as synthetic media, like MS media. And semi-synthetic media, the chemical composition of media is partially known as semi-synthetic media, such as PDA, that is potato dextrose agar. Another criteria for classification is on the basis of consistency. So if a liquid media, sometimes referred as broth, the media which provides film surface on which cells can grow for discrete colonies, these are called as solid media. So here in the solid media, what is the difference between a liquid media and solid media? In a liquid media, which is also known as broth, it lacks agar or solidifying agent that is present in a solid media. I have here mentioned different type of solidifying agents such as agar-agar, gelatin or silica gel. So what is agar-agar? So there are few points to remember. Agar-agar is the most common solidifying agent used at concentration of 1 to 2% to make a solid agar media. It is preferred because of its properties like it melts around 98 degrees Celsius and solidify at below 42 degrees Celsius. Agar is chiefly used as an ingredient in desserts. Agar is an unbranched polysaccharide obtained from the cell membrane of some species of red algae such as genera of Geladium, Gracilaria or seaweeds. Likewise, gelatin. It is a protein obtained from collagen of animals byproduct, but it has low melting temperature around 50 degrees Celsius. So next is semi-solid media. The media which exhibit a clot-like consistency at room temperature are called as semi-solid media. They contain a solidifying agent agar-agar at 0.2 to 0.5% that thickens them as custard-like consistency but does not produce a film substrate. These are used to determine the motility of bacteria and for cultivation of microorganic bacteria. So another based on the utility or purpose. Simple media and enriched media, selective media. A simple media is a basal media, basically that supports most non-fastidious bacteria like peptone water, nutrition broth, nutrition agar, etc. And enriched media, and this addition of extra nutrition in the form of different substrate like egg yolk, serum, blood, etc. to the basal media makes them enriched media. For example, a differential media, certain reagents or supplements when incorporated into culture media, they may allow differentiation of various kinds of bacteria. This type of media is known as differential media. And the last one is selective media. This media provides nutrients that enhance the growth and predominance of a particular type of bacterium and do not promote other type of organism that may be present in is known as selective media, like selective media for trichoderma. So coming to the objective of today's lecture, preparation of culture media. So here we will discuss two culture media. One is for fungus and another one is for bacterial microorganism. So for fungus microbes we use potato dextrose. So you can see the material required for preparation of a liter of PDA media. You require 200 gram peeled potato, 20 grams dextrose, 20 gram agar, and conical flask of 250 ml preferably. Also you required autoclave for sterilization, non-absorbent cotton, measuring cylinder, muslin cloth, knife, etc. You can see these are the chemical used and the composition of PDA, that is dextrose, agar. So what is the procedure? First you take 200 gram of potato, wash them thoroughly and cut the potato into thin slices and boil into 500 ml of distilled water till they become soft. Filter the content of the beaker through muslin cloth and squeeze out all the liquid. Add 20 gram of dextrose dissolved in water to this extract. It will give you 1 liter PDA media. So another media is culture media that for bacteria, that is nutrient agar. The composition of nutrient agar is peptone, it is used 5 gram, beef extract, it is used 3 gram, agar agar 20 gram, distilled water 1 liter, and the other miscellaneous materials. Here you can see a peptone, beef extract, agar agar. So what is the procedure? Dissolve the weighted amount of peptone and beef extract into 1 liter of water. Weigh 20 gram of agar and completely dissolved. Stir constantly and cool at room temperature. Adjust the pH of the medium to 7 by adding 0.1 HCl or 0.1 normal NaOH. Dispense 200 ml each into conical flask. Here you need not to filter the liquid through muslin cloth. The point number 5 is miswritten here. Plug the flask with non-absorbent cotton and sterilize at 121 degrees Celsius at 15 psi for 15 minutes. Allow the autoclave to cool, remove the conical flask and store at room temperature. So these are the two media, the procedure of media. So now what are the questions one should keep in mind? First, don't pour the media over 2/3 of flask capacity because it will be problematic when you reboil the media for further use. Next, cotton plug must be loose while autoclaving. Too tight cotton plug may create extra pressure and the flask may get break. Next is don't pour media to petri plate when the media is too hot, since it produces condensation of water or undesired of petri plate lid and thus can fall on the agar surface and may lead to contamination and spreading of colonies. So wait till the media get lukewarm, and the simple method when you're able to handle the flask easily, so that at that temperature you can start pouring. While pouring, pour media quickly to avoid contamination by air pores and close the lid down as soon as possible. Perform the pouring of media in inoculation chamber fitted with ultraviolet lamp and filtered air. And also it should be performed near